All right, so real quickly, I have 6 internet students who have not logged on into their Blackboard account at all, and they will be dropped from the course if they do not follow. There are instructions for internet students on how they do attendance. So if you're an internet student, read through your Blackboard account, and it tells you what you need to do for attendance, because if you do not log in, it will think that you are not in this course, and you will be dropped. For the regular people, you guys are fine, as long as you've attended at least once. And but internet students, you need to do that clicker attendance in order to show that you have registered and stayed in the class and all that good stuff. Now, for the rest of you guys, did you guys do the homework assignment? Did you play with cyanide? Not for reals, right? Okay. So let's try to do that cyanide problem real quick. So with cyanide, we said that it was HCN, right? And what do you guys know about hydrogen? How many bonds can it make? One single bond, right? It only has one electron and it needs a buddy. So we know that it can make one bond. And what did you guys know about nitrogen? Because we did that one before. It has 2 paired electrons, but it has how many unpaired electrons in its valence shell, the outer shell? 3. So that means it can make how many bonds? 3. So 1, 2, 3, a triple covalent bond. So if you didn't know about carbon, which I did mention, carbon had how many unpaired electrons in its shell, its valence shell? It had 4 unpaired electrons. So another way that you could, just for your own brain purposes, so you knew hydrogen had 1, you knew nitrogen had 3 unpaired ones, and you knew carbon needed 4. So here's 1, 2, 3, 4. So it makes them look like little dumbbells, but sometimes that helps if you need to see like the dots, not just the lines. But you guys know that a single line means it's a what kind of bond? It's a covalent bond. If it's a single line, it's a single covalent, double covalent, or you got it triple covalent if it's 3 lines, right? So that's what it should have looked like. H, single covalent bond to a carbon, triple covalent bond to a nitrogen. And if you drew out the electron shell, the valence shell, to see that, that's how I'm kind of helping depict the little dots there, just to help you out if you needed to visualize it a little more. So let's see. Any questions on that? Not so much. And I think we ended with, let's see, we talked about our bonding patterns. We talked about the special things about water that make it super unique and awesome. Yes. And we finished up briefly by talking about, if we scroll down a little bit, we talked about cushioning was the very last thing we were talking about with water, because your CSF, or cerebral spinal fluid, has a lot of water in it. It has some ions and other things as well, and that helps cushion your brain, right? If you had some sort of traumatic injury to your head, either something smacking you in the head or you falling or whatever, and helping it not knock around so much. So that leads us now talking about acids and bases. So with acid and base. So, all right, so acid is a proton donor. And what we mean by that is HCl, or hydrochloric acid, is considered a strong acid. So what it means is if you put acid in water, it would immediately want to dissociate or come apart. So hydrogen would separate from the chloride, and you'd have ions then. That's why I'm showing you H plus and a Cl minus, right? And we know it's ionic because that's a full charge, not the little partial charge with the delta sign that I showed you a little bit last time. So a strong acid means it wants to dissociate or come apart easily when you added water. If it were a weak acid, it wouldn't really want to come apart. It would stay as that molecule, wouldn't want to separate. Okay, that's what a weak versus strong acid means. So that hydrogen ion by itself, that H plus, another name for it is a proton. Okay, so you'll hear us sometimes call it protons, you'll hear it as a hydrogen ion. And that H plus, is that a cation or an anion? Cation. Remember cat with a T plus sign, or chew it up, right? And then Cl minus must be an anion. Okay, I'm mentioning this to review for you, as well as you're going to see this a lot over and over again. If we talk about a base, okay, that would be a proton acceptor. So it means it would want to accept that hydrogen ion or a proton, okay? So in this case, we put NH3, okay, which is ammonia, and if we put that in an acidic solution, it will decrease the acidic solution because it would create the ammonium ion, which is the NH4 plus. And ammonia is basic because it took up a proton. So that NH3 took up the proton, so it was a proton acceptor, that's why it's a base, and then it created the ammonium ion with an M. And the ammonium ion is a cation or anion? It's a cation because it has a positive charge to it, right? So another thing too is that we do this process all the time. This has to do with your urine. So you'll see this equation later on down the road. pH is something that we use to measure the proton or hydrogen ion concentration. So basically it's a pH scale from 0 to 14. 0 or closer to 0 would mean it's more acidic, or another way of saying that is less basic. And if we're closer to the 14 end of the scale, so 13, 14, it means we are the least acidic or more basic, correct? And oftentimes, sometimes like in chemistry, you might talk about it more as acid and base. And a lot of times in here, you'll hear us just say it's more acidic and less acidic. But I want you guys to also understand acidic versus basic or neutral also. So where do you think neutral might be on the 0 to 14 scale? 7. So 7 means it's not acidic, it's not basic. If I said you're a pH of 6, you are more acidic. If I said you're a pH of 9, you are more basic or less acidic, right? And in 228, you're going to learn that our pH for our body, our neutral pH is 7.4. We're not exactly neutral. We are what? We're slightly basic, right? So this is like where I, talk about sci-fi stuff, and in Aliens, they had acidic blood or spit and stuff, right? I mean, where do you think they get this stuff from? So for us, 7.4 is neutral for us, okay? But if you're talking straight pH scale, our bodies are technically slightly basic or alkaline a little bit. That's another term, I guess you could say. So for neutralization, okay, so if we take HCl or hydrochloric acid, which is a strong acid, we said, right? And then we take sodium hydroxide or NaOH, okay? And NaOH is a strong base. So we have a strong acid plus a strong base. What we end up getting is NaCl. Anybody know what that is? Table salt, right? Plus HOH. Anybody know what that is? It's another way of saying H2O, right? Water, okay? It's just spelled out a little different. Or HOA. No, I'm just kidding. So basically, if we take a strong acid and a strong base, it neutralizes it. You end up with a pH of 7 then for neutral, right? And this does play a role in our physiology, and especially, even though I keep saying to you guys, we'll see more of this in 228, it's because what you guys are learning here are your building blocks as you get more and more information this semester, and then you're definitely going to see it again in 228. And so if you guys get this under your belt now, it makes more sense by the time you see it again. That's why we bring this up. If we look at salts, another name for salts is electrolytes. So you guys hear like when people sweat a lot and they say, oh, you should drink some Gatorade because it has a bunch of electrolytes in it. Because when you sweat, you're not just losing water, you're also losing your salts, like sodium, potassium, chloride, right? These are all ions from ionic compounds. Magnesium, calcium, those are all parts of salts and stuff too, right? So sodium and potassium are cations or anions. You guys are going to get tired of me grilling you, huh? And chloride would be an anion. All right, see? Nice and easy. All right, What else do we want to say? So if it's a hot day and you're sweating a lot, you should just drink water, or should you drink something else? Gatorade may not be the best choice, but Gatorade has a little bit of glucose in it, and it also has some salts in it. So it helps replace some of the salts or electrolytes that you were losing. So people that get leg cramps or muscle cramps after exercising or marathoning, or maybe they don't normally exercise and all of a sudden they started exercising, it's probably because they sweated out a bunch of their salts and they need to replenish that so they can get their muscle physiology back on track, right? And now let's look at water mixtures. So no chemical changes of components, all right? So basically we're saying that it's easily physically regain them. So you're not bonding something, okay? It's kind of like things are mixed in a solution, but you're not creating new bonds, is what I'm saying here. So if we look at solutions, or before I say solutions, let's talk about solvent. So solvents are substances usually in the greatest quantity, and it's typically going to be fluid, or most of the time we're talking about water, because water in the fluid phase. And what did we say about water? Water is the universal solvent, okay? And if we talk about solutes, what we're saying is these are substances typically in the lesser amount, and these are also typically going to be more your solid or particles. So that's why I wrote particles there. So how does this come into our water mixture? So if we look at the term solutions, this is a homogeneous mixture of two or more components. So homogeneous or homogeneous, homo meaning the same. So you're saying it looks the same throughout. So it wouldn't matter where you took a sample of a beaker of this solution, it would look the same throughout. You couldn't see two different things in there, okay? So a mixture is the same no matter where you sample it. Particles are small, so they're small enough that they don't end up scattering the light, so it looks pretty much transparent. So it kind of looks like a clear glass of water, but it might be a glass of salt water, or it might be a glass of sugar water, but it looks the same. You're not seeing particles sitting on the bottom. So I don't mean like you put so much sugar that it's super saturated and you got sugar sitting on the bottom. I mean, it's totally mixed in and it looks like a glass of water. That would be a solution, okay? If you have a colloid, this is heterogeneous, hetero meaning different. So you can see slightly different. So heterogeneous mixture that appears milky. So it's similar to a solution. So SOLN is my abbreviation for solution. And the X with a line in it means accept. So that's a shorthand or medical terminology for accept. So it's similar to a solution, except the particles are big enough to scatter light. And that's why it looks a little milky, or in this case, translucent. So it's not clear, but you can see that there's stuff in it. It's not just water, right? If we talk about a suspension, this is a heterogeneous mixture. So heterogeneous meaning different. Heterogeneous mixture, this time it's particles large enough to settle out over time. So you could think of this as if you went out to the ocean and you scooped up, near the beach, a little jar, bless you, and you're holding it, and at first it might look like it's all mixed up together. And if you let it sit for a little bit, what settles out into the bottom? A bunch of sand, right? So the difference is that this stuff settles out versus solution and colloid, it doesn't settle out. So I put a little thing for A and B to say particles for a solution and colloid. Particles do not settle out for solution or colloids. And suspension, though, it does settle out over time. Okay. So a suspension, you could think of this as like sand and water. Okay. Another suspension that will make more sense to you guys is blood. So when you first take blood out of somebody in the tube, it kind of looks like it's one solution, right? But if you leave it sitting out over time, it starts to separate. And so that tells you that it's a suspension because the solid particles, the cells and cell debris is settling out, and the liquid portion is sitting up on top. So sand and water or blood. I know it seems silly, blood, I just put BLD, like skip the double O's, but whatever you want to write it as. A colloid, for example, so this is where it doesn't settle out. That would be something more like Jello, okay? It looks the same throughout, but it's not completely clear. Or you could even think like skim milk. And if we go back up to a solution, that would be something like sugar water or salt water, where basically it looks homogeneous. It looks like it's all the same thing, but it's not settling out or anything. So an emulsion is like a subset of a suspension. So an emulsion would be, it says water mixed with a nonpolar substance and agitated. Bless you. So that means that once it sits for a while, it's going to do what? It's going to separate, and you're going to see two different levels of something, right? So if one's polar and one's nonpolar, if we were using water and fat, you guys know those don't really mix. Like you could shake it up and at first it looks like they're kind of mixing, but you wait just a couple seconds and then you start to see the fat bubble up separate from the water, right? So like a vinaigrette would be vinegar, which is polar like water. So somebody might add vinegar and oil together for their salad dressing, and they'd shake it up and pour it right on. But if they shook it up and then let it sit for a while, and then you come to sit to eat and you pour it off without shaking it again, you're going to get just the oil part probably or something, right? So that would be an emulsion, would be like a salad dressing, I'll put. Not like a creamy one, but like a vinegar / oil one, vinaigrette. All right, because they don't mix. So then if we look at the picture here, so they're giving you an idea. So we're talking about, here was that emulsion by itself. There you're showing you the separation of oil and water. They kind of mix when it's in motion, but when you let it settle out, they separate. Here was the mixture. So we had solution, colloid, and suspension. We said a solution, that would even be like mineral water. Salt water, minerals, right? Same thing. Colloid would be like a gelatin, like the Jello like we talked about. And then blood would be a suspension or sand and water, so it ends up separating. So here, when they first take the blood out, here you're seeing the plasma, the liquid portion of it, separating out from the white blood cells and then the red blood cells and platelets. So that's pretty much all that is about. So it's basically go back, again, review the terminology, try to use it, get it stuck in your head. That way you have less to worry about when you're trying to study for the exam. Now we get to get into chemical reactions, and it says they occur when forming or breaking bonds. So forming, another word for forming would be making, right? So we're either making a bond or we're breaking a bond, okay? And either way, it's a reaction. So chemical reactions are the symbolic form of a chemical equation is the symbolic form of a chemical reaction. It's just saying, how do we write it on paper to show what's happening, right? So reactants, it says, are substances entering the reaction. So typically, I'm just going to use the letter R for reactant. They would be on this side of the arrow. I don't just mean the left or the right, but for basic purposes, I have it on the left side of my arrow. The arrow is pointing away from it. So we're going to somewhere else, right? And if I say a product is the substance or substances formed by a chemical reaction, it would mean that products, and I'll just put that as a P, would be on that arrow side of the arrow, okay? So typically you have a reactant can either be broken down into product or products, or you have a reactant, you might have more than one reactant coming together to form one product. You might have one reactant getting broken down into two products. So now we're going to look at different patterns of what could happen with reactions. But a chemical equation basically describes these reactions. So for example, so I could also say this, right? Saying the same thing. Just look at where your arrow is pointing is what we're talking about here. So if I say the chemical reaction, let me see if I can add a space here. All right. So if I say H2O plus CO2, and here's an arrow, and I have H2CO3, okay? Is H2O a reactant or product? It's a reactant because of the side of the arrow that it's on. And is CO2 a reactant or product? It's a reactant. So both H2O and CO2 are reactants in this case. And H2CO3 is carbonic acid. And is it a reactant or product? It's a product because it's on the other side of that arrow. Okay. So this reaction is actually how we get rid of CO2 in our own body physiology. So our cells are making CO2 all the time, right? Because what do we end up doing? We breathe in CO2, we breathe in O2 and we breathe out CO2. You breathe in too much CO2, that's not good. Sorry. So we take in O2, your cells utilize it, okay, and your cells give off CO2, right? Carbon dioxide. So we have to get rid of this carbon dioxide somehow. And what we do is we squish it together, do a reaction, put it together with water, and we form carbonic acid, okay? Then we can make that travel in our body and get it up to the lungs. And when we get to the lungs, we can separate it again and break down H2CO3 into H2O and CO2. And then that CO2 can be breathed out. So this is why I was saying reactions can go either way. And sometimes you'll see things written as R. Well, that's like a half arrow. We'll go to P. And in other conditions, P will go back to R. But if P goes back to R, now that's an R going to a P, technically, right? Just follow the way the arrows are going. Think about it again later. Go with the main part first. That'll make the most sense. Let's see, what else do I want to say? So carbonic acid, it's a weak acid, meaning it doesn't want to separate well, so that's why it stays together. And anyway, patterns of chemical reactions. There are 4 patterns that I need you to learn. So if we look at, we have a synthesis reaction, a decomposition reaction, an exchange reaction, and an oxidation reduction or redox reaction, okay? So with the synthesis reaction, another name for that would be a combination reaction. And I abbreviate reaction as RXN, okay? Anytime you see an arrow is indicating reaction, essentially. So R going to P, that arrow between R and P means reaction. So this means that chemical bonds are formed. Because synthesis means to make or synthesize, right? So what we're saying here is our generic equation of A plus B, A and B separate are what? Reactants. And they're going to form the product AB. And we didn't draw a line between them, so technically you don't know if this is an ionic bond or a covalent bond. If it were technically an ionic bond, you'd probably see some positive and negatives up there. If it were a covalent bond and we were being super specific, we might show the dash line, the single covalent bond between them or something. But we just know that it's made a bond. That's all we know from that. And so there's one reaction you guys have already learned that was a synthesis reaction. If water was used, what do we call that one? No, it's the opposite of that one. Dehydration synthesis. You're on the right track though. We're coming up to that. So dehydration synthesis would be a synthesis reaction, right? And one of the products of that would have been water, because you would have had like an H and an OH, an H off the A and an OH off the B, and when you stick A and B together, you pulled off that H and an OH, so you've actually formed water. A decomposition reaction is the opposite of a synthesis. So you could think decomposition like a decomposing body, decomposing vegetation, something like that. So this is where chemical bonds are broken. Okay. And so they're showing you AB is what now? AB is a reactant. It's getting broken down into two products, A plus B. Okay. So would water be on the reactant side or the product side if this was hydrolysis? Right. It would be a reactant because you added it into the equation to break up AB. And then one of the H of the H2O would go on the A and the OH might go on the B. So you've used water to help break up AB that was together. So it would end up being like H2O plus AB. So water and AB end up being your reactants, and what else do I want to say? And it's a hydrolysis reaction. Okay. So let's see, an exchange reaction. This actually includes both decomposition and synthesis, and it's in that order. Okay, so it's a decomposition reaction and then a synthesis reaction. So the equation, the generic equation we gave you there was AB plus CD yields or gives you AD plus BC. So what really has to happen here is if you had AB plus CD first has to get broken down, that's why it's a decomposition reaction first, into A plus B plus C plus D. And then it becomes a synthesis reaction to exchange or rearrange things around and form bonds again to give you the AD plus BC part of it. So this was the decomposition part of it, and this part was the synthesis part of the reaction because you exchanged it and reformed it. So the nice thing is a lot of times the terminology used is telling you exactly what's going on, so don't overthink this stuff on exams, okay? Next, we get to learn about oxidative reduction reactions or redox, okay? All this is electron exchange reactions. So little e means electron exchange reactions. So it's basically shifting an electron from one molecule to another. And we do this a lot to transfer things around, and it ends up helping us later when we learn about metabolic regulation, when we learn about metabolism in 228. You're going to see a lot of these redox reactions then. So basically, if we look at an electron donor, means that it is oxidized, and an electron acceptor means that it's reduced. So what we're saying is if something donates an electron, if it's donating it, it's like letting it go, right? It's letting it leave. If an electron is leaving, okay, then it's become oxidized. So if we said you have Na and Cl, right? And remember when he lost, Cl stole his electron to become an ion, right? So you had Na plus Cl, and you ended up with Na plus ionically bound to the Cl minus. Cl stole his electron, so he became more negative. So if you're the one that lost the electron, it means you were oxidized. So if sodium lost an electron to chloride, it means sodium was oxidized. And if Cl was the one that received the electron, it means Cl was reduced. Okay? So another way of thinking of this is, one, you can just learn a mnemonic. Okay, so the one that I like to use is LEO and GER. LEO is lose electron means you're oxidized. Lose electron, oxidized. GER is gain electron, reduced. So it's a super easy way to remember who is reduced and who is oxidized. LEO and GER. If you lost your electron, you're the oxidized one. If you've gained the electron, you're the reduced ion. And the other chemistry way of knowing it is because you have gained the electron, you've been reduced because you're less positive, which I don't know if that just throws you guys off more or not. I just like Leo and Gur because it's super easy, right? Lose electron, you're oxidized. All right, so now moving on, energy flow in chemical reactions. So this is another way of describing what's happening. Some of this you can think of as that, potential energy and kinetic energy. This kind of goes in line with some of that. But basically, an endergonic reaction means that energy is absorbed, and an exergonic means energy is released. So which one of those two do you think means that a bond was broken? Exergonic. So bonds broken. And we mentioned that before where I said, hey, if we break this off, we release tons of energy. That's what we're talking about. It was an exergonic reaction. So the opposite must be true of endergonic. So if it takes energy to build something, that makes sense, right? Anytime you're trying to build something, you have to put energy in to do it. So it absorbs or takes energy to do an endergonic. So all synthesis reactions are endergonic. All breakdown reactions are going to be exergonic. Okay? So I'm going to put endergonic means makes bonds. So you can go back and go up to these reactions we talked about here. Synthesis reaction, dehydration synthesis would be ender or exergonic. Endergonic. And decomposition or hydrolysis reactions are all going to be exergonic. So go back and play with that a little bit. Obviously, exchange reaction would be a little bit of both, depending on what part of the reaction you're at, right? So play with those a little bit. Now, let's look at factors that influence chemical reaction rates. So this is kind of talking about what is more likely to make something occur or not, right? So things that speed up or slow down the reaction rate. How do you think temperature would affect a reaction rate? The hotter it is, the faster it's going to go. Remember when I talked about fevers, right? We're trying to kill things before we kill ourselves. Because if your temperature goes too high, you're going to break things down. And we'll talk about that in a second. So if we increase, and I'm just going to put T for temperature, it will also increase the reaction rate. So then the opposite would be true. If we cool things down really, really low, then we do what to the reaction rate? Slow it down. So this is like when somebody falls into the ice cold river and you pull them out and somehow miraculously you're able to revive them. They died, but you brought them back because their metabolism slowed down so much that it kind of protected things, and then you brought them back to life. May not always happen. Probably wouldn't happen if it were like 90-degree water. You might be dead for good. So what about the concentration of particles? So this is concentration means like the amount. It'll go up. So the more reactants you have, the more stuff you have, the more likely it can bang into something else and form another bond or do whatever it's supposed to do in this reaction. So I'm just going to put, oftentimes when you see concentration, it has brackets around it, and I'll just put C because we're being generic with a concentration of something. So we increase the concentration of our reactants, then we would also increase the reaction rate, and vice versa. If you have very little reactants, a low concentration, it's less likely to bang into something to form the new bonds or break down, okay? What do you guys think about particle size? What would that do to reaction rate? If you're a ginormous particle or you're a teeny tiny particle? It's going to go down. If you're a huge particle, think about being really slow. And if you're a teeny tiny particle, like an electron, you're going to zip around like a little yellow jacket. So if you're tiny and zipping around really fast, you're more likely to bang into other stuff. If you're ginormous and huge, just think that you're kind of slow and it takes you a while to maneuver across the room. Okay, so increased size is actually an inverse proportion, inverse relationship. So an increased size would actually decrease the reaction rate. Okay. On catalysts, these increase the rate of reactions. So catalysts basically help bring reactant or reactants together. So if you have two reactants coming together, it'll help put them together so that they're more likely to make their bond. So this is their job. So catalysts bring reactants together. I'm just going to put TOG for together. They are not changed by the reaction. So not, and I use the triangle for change, not changed by the reaction. What that means is a catalyst is not a reactant and a catalyst is not a product. It's just like a helper buddy. Comes in, helps bring things together, but it's not really part of the reaction. It doesn't get broken down and then it becomes part of the product part of it. It just comes in, helps them come together, goes away, and it gets recycled. So it can do it again and again and again. Okay? And for our purposes, all of our biological catalysts are enzymes, which means for us, they're all proteins. Okay? So enzymes, and I just put ENZ, enzymes, or an enzyme, is a biological catalyst. Okay? So our enzymes are proteins, and it's another reason why, okay, if we need these catalysts to help make sure that certain reactions occur, and our catalysts are all proteins, and if we have a fever, and fevers break things down, like look at egg white, you know, when you put that in the pan, you heat it up, and it coagulates it, it turns it solid white, right? You can't get it back to that thin, clear, translucent egg white, right? So you've cooked it. So it's kind of the same thing with the fever. If you have a fever that gets too high and for too long, it starts breaking down your proteins. If it breaks down your proteins, thus your catalysts, your enzymes, certain reactions will not be able to occur. And you've damaged it, you're done. So that's why fever's too long, too high, not good. All right, let's get into biochemistry. Woohoo! I love biochemistry because that was my major. You guys are going to love it later too. Probably not. I don't know. You might. All right, so inorganic compounds mean that they lack carbon. Okay? So give me some examples of some inorganic compounds that we've already talked about in lectures so far. Water, yes. Does not have a carbon in it. And it's a compound. Salt, NaCl, very good. How about hydrochloric acid? Right? Now there are two exceptions to this rule. They have a carbon in it, which is carbon monoxide and carbon dioxide. These are our only carbon exceptions. You'll understand why in just a second. So for organic compounds, that means that all carbons, it contains carbons and hydrogen. That's why CO and CO2 are considered inorganic. They're not a hydrocarbon. They don't have hydrogens and carbons. Okay, so that's why CO and CO2 are an exception for an inorganic compound. All the other carbons with hydrogens attached mean that they're organic compounds. So another name for this is hydrocarbons. Okay. So we're going to look at our important organic macromolecules so that we can understand how cells are built and put together. So if we look at some of our macromolecules, for example, we have carbohydrates. So carbo for carbons, hydrates, it's a hydrocarbon. It means it's got tons of carbons with lots of hydrogens around it. Okay. And so carbohydrates are basically our sugars and starches. Most of you guys have a general idea of what a carbohydrate is, and you hear people say, I'm on a low-carb diet. It doesn't mean they're not eating zero carbs. It means they're probably eating some carbs. They're just not eating, you know, obvious sugars or simple carbohydrates like boxed foods and things like that, okay? They're still eating starches and carbs. They just don't always realize it. So this is actually our main source for our cellular fuel, and it contains carbons, hydrogens, and oxygen. And the neat thing is that for every carbon that we have, there are 2 hydrogens. So it's actually 1 carbon per 2 hydrogens per 1 oxygen when it gets put together. So that's your base structure of a carbohydrate, CH2O. Now, let's see. Also, let's put cellular fuel, because it is. And they dissolve easily in water. So what does that tell you that carbohydrates? No, it's not. Yes, it is. If they dissolve easily in water, what does that mean? It's got to be polar. It's got to have some polarity to it, right? So also polar substances. This is what happens when you get sick. I apologize if you guys tried to come see me yesterday. I ended up with the stomach flu. I can't catch a break today. All right, so that means it dissolves readily in water, right? So let's say you take a Pringles chip and you pour water on it and you let it sit for a while. It's going to end up breaking it down a little bit. Not as well as the other enzymes you have in your mouth, but it will start to dissolve it a little, right? If you had sugar sitting there or salt sitting on the table and you pour water on it, it's going to dissolve it a little bit, right? So sugars are part of your carbohydrate, not the salt part. I was just saying it would dissolve it. Okay, so now if we look at subdivisions, if you will, of carbohydrates, we can talk about a monosaccharide. Anything with saccharide in it tells you it's a sugar. Mono means 1. So we're saying a single sugar, and this is also another name for this would be a monomer. Monomer. Monomer, think of this as 1 Lego block. This is the building block of a larger molecule. So a single one, a single monomer, or 1 single monosaccharide. And the one we'll talk about the most is glucose. And you do need to memorize its molecular formula, which is if I tell you it has 6 carbons in it, how many hydrogens does it have? Look at the base formula. 12. And how many oxygens then? 6. Okay. So if you're not sure, if we said there were 6 carbons, you go back to the base formula we said was CH2O. So for every carbon, there are 2 hydrogens and 1 oxygen. That's why it's C6H12O6. So you don't have to memorize this picture here, but you do need to memorize this formula there. That tells you it's glucose, it's a monomer, it's a monosaccharide, okay? We can link glucose to others and create a disaccharide, and we can link it many, many times and get a polysaccharide, okay? But real quick, still while we're under monosaccharide, again, this is our building block for larger things like carbohydrates. So we'll put monomer is a building block. So think of 1 Lego would be a glucose. Many Legos put together give you the carbohydrate. There are other monosaccharides. You don't have to memorize these guys, but you might want to be a little familiar with just the terms. So there's MERMs, galactose, fructose, ribose, deoxyribose. So fructose is like fruit sugars, what you find in a fruit. Galactose you're going to find like in milk product, okay? Ribose and deoxyribose, anybody sound a little familiar to you? DNA and RNA. So what? We have sugar in there? Yes, we do. So you're going to see that as we build our DNA and RNA molecule, okay? So that's our monosaccharide. If we look at disaccharide, di means 2. So we're saying 2 sugars linked together, okay? And so an example of this would be if you had sucrose is a glucose linked to a fructose. Okay, so there's your sucrose there. Again, you don't have to memorize the pictures, but you can see that you have 2 sugars linked together by a what kind of bond? Generic. covalent bond, right? And sucrose is table sugar. So if you have like the little sugar dish on your table, that's sucrose. Lactose is a milk sugar. Maltose is a malt sugar. I'm sorry. And alcohol, yeah, for beer. There you go, alcohol sugar. How do you know that? No, I'm kidding. I'm kidding. I'm teasing. Just kidding. So that's your disaccharide. Another name that you might see is dimer, is another name for two being together. So you might see dimer. Okay. And then they are covalently bound together. If we look at poly, poly means many. So you're saying many sugars linked together. So this is a long chain of simple sugars. So for example, if you have starch, starch is the glucose polymer of plants. So a polymer just means when many are linked together. So starch is the plant polymer of linking many glucose or glucai together. I don't think glucai is a word, but you know. So starch equals plant storage, because when we link things together, we're storing it for later. So it's a plant storage or glucose polymer. And guess what? We can do this too. So not just plants do this. With plants, this would be like eating a potato chip. You're eating their starch. You're eating many glucoses that are all together. Now your body will take that and break that starch molecule down and break off each of those glucoses or other sugars that it might be made out of. And then you'll use what you want, and then we will store it for later use for us, energy storage. So if we store it, we call it glycogen. So glycogen is the mammal or human, but other mammals do it too, mammal storage of glucose polymer. And we're just saying glucose just to keep it easy, but there could be other sugars involved and stuff. So again, this is our storage form of glucose. It's a large, ginormous macromolecule. So when you're saying starch, you're always talking about a polysaccharide. You're not really talking about a single glucose molecule, because starch is a large molecule. That's the difference here, really. If we look at our next macromolecule, so we had carbohydrates. Now we can talk about lipids. Lipids are organics that do not dissolve in water. So is it polar or non-polar? Non-polar. All right. It basically means less oxygen means that there are less hydrogen bonds, which means that the lipids do not like water. They're hydrophobic. Because remember when we said hydrogen was linked to an oxygen, and oxygen was so huge and it had that partial charge, it was pulling the electrons towards it. Well, if you don't have something to pull those electrons and they're sharing the electrons equally, they are just a non-polar covalent bond. That's what lipids are, non-polar, but they're still covalent bonds. That's why you don't have these partial charges. If you don't have partial charges, water's not going to be attracted to it because it's not a polar molecule. So basically, we're saying it's non-polar, which makes sense because you guys know fat doesn't dissolve in water. It stays separated, right? And so water and fat don't mix. Because of that, it is hydrophobic. So phobic means you have a fear of water. Fear of water. It's probably not really afraid of water. It just doesn't like it. So we're saying because water is polar, and we're saying fats are not, thus do not mix. They stay separated. So they have carbons, hydrogens, and an oxygen, but it's not as many oxygens as like the carbohydrates, which is why they're non-polar. Not many O's, thus why they're non-polar. So if we look at the term neutral fat, okay, neutral fat we're saying is a triglyceride. So another way of abbreviating triglyceride, you'll just see TG sometimes. Okay, this is our storage form of fat. So when you guys look at an adipose cell under the microscope in lab this week or next week, it kind of looks like chicken wire. So the nucleus is squished right up against the cell membrane because what was stored inside of that cell were the storage form of fat, or in this case, triglycerides. So triglyceride is a monomer. This is also the most common fat found in our bodies because it's our storage form. So triglycerides are the most common fat found in us. It's very nonpolar and most common in us. Okay. It is a monomer. So monomer means like the single building block of it. The monomer is one glycerol, which is the backbone. So one glycerol molecule plus 3 fatty acid chains. And from here on out, I'm going to say fatty acid is just Fa to be simple, okay? So the triglycerides are fatty acid chains, which are hydrocarbon chains, okay? And what we mean by that is many, many carbons with many, many hydrogens around it. And they are always an even number of carbons in the chain. So always an even number of carbons in the hydrocarbon chain. Okay. So we have 2 types of fatty acids, or general categories of fatty acids, I should say. We have saturated and unsaturated fatty acids. And this has everything to do with whether there are double bonds in the fatty acid chain or not. So a saturated fat means that there are no double bonds between the carbons in the fatty acid chain. Okay? So another term that you could use with a saturated fat would be hydrogenated. So essentially what saturated means is it's saturated with hydrogens, and that's what hydrogenated means, is tons of hydrogens all the way around it. So another name is hydrogenated fat. Okay? Maximal hydrogens, saturated with hydrogens. So if it's saturated with hydrogens, there are no double bonds. If it's an unsaturated fat, it means it does have double bonds occurring between the carbons of the fatty acid chain. And another name for that would be dehydrogenated because there's less hydrogen. So dehydrogenated. All right. So basically, if we look at the picture down here, what we're showing you is if they take, in the pink box here is one glycerol molecule. The whole pink box is a glycerol molecule. So that's the glycerol backbone. And then we have 1, 2, 3 fatty acid chains linked to it. And I want you to look at where they have the pink box and the gray box next to each other. And they're showing you how they're linking them together to finish forming the triglyceride below. So what kind of reaction do you think this is to link the fatty acid to the glycerol backbone to create that bond? Dehydration synthesis. We're synthesizing because we're building a larger molecule, right? Dehydration will remind you because look, it formed a water, meaning water was removed from between those two molecules. Okay. So the reactants would have been glycerol plus fatty acid would have yielded, after you add your other fatty acids also, product would have been a triglyceride, technically. All right. So of these 3 chains that you see here that I numbered 1, 2, and 3, which one or ones would be a saturated fatty acid? Number 2, because number 2 has no double bonds in the fatty acid chain and it's saturated with hydrogens. And what that means is if you look at, so which ones are the unsaturated fats? Number 1 and number 3 are unsaturated. So if you look here, This carbon and this carbon, we're missing the hydrogens, right? If we're looking at the double bond area. So it has a double bond, C double bond C, right here. And because that extra electron would have gone to an H, instead it went to the double bond to the other C. That's why it's unsaturated. You removed a hydrogen and created a double bond between the two C's. And so same thing down here. Here's your other carbon-carbon double bond. Thus, there are two hydrogens missing. We don't have one here or here. But if you actually do the valence electron shell, you would see where those carbons or those electrons are between the C's then to form that double bond. If you're not sure, if what I'm saying you're going, I have no idea what she's talking about right now, I want you to draw out what the carbon to carbon and the hydrogens around it would look like with the valence shell, and it'll make more sense. If after you've done that you still don't understand, then come talk to me, okay? Because it will make sense if you see it. Let's see. So one kind of fat, saturated versus unsaturated, would be more commonly found as a liquid, and one would be more commonly found as a solid. So if I tell you that when you see those double bonds, it actually creates a kink in the chain. So this picture is showing like the hydrocarbon chain just goes straight across. But it would really be like, here's your glycerol backbone, and here's 1, 2, 3 fatty acid, and here's a double bond, and it would kink up all of a sudden. And this middle one didn't have a double bond, so it stays straight, and maybe this one kinks down because of that double bond, right? So those double bonds create a kink in it. If it was a completely saturated, if all 3 fatty acids were completely saturated, then it would look like this, right? So which one do you think would stack more uniformly next to another one? The saturated fats. And because they stack, they're not getting the kinks. It's like there's less, let's pretend like they're logs, there's less air pockets between them and stuff. So if you can stack something more tightly together, does it make sense it might be more solid? Versus if you have kinks, you have more space in between and stuff. So a saturated fat, like butter, it's solid at room temperature, right? Olive oil has some double bonds in it. So it's liquid at room temperature. That's the difference between saturated and unsaturated. And we won't go into deeper details, but you know, like if you've heard the term like omega-3 fatty acid and omega-6 fatty acids, right? The omega is just telling you which carbon has the double bond. If something is a polyunsaturated fat, it's healthier because it has more double bonds in it. It won't stack as tightly. That's all they're telling you. But you're going to have patients that come in and they have all the cool lingo down, but they have no idea what it means, right? You guys have all heard some of that term, and some of it you're like, yeah, I know what that means. Did you really? So when patients talk to you, this is important to keep in mind. Just because they're using the right terminology does not mean they understand what you're talking about to them. That's why you need to make sure that you explain things, what we say like in layman's terms. Make sure you explain it in a way they understand. They don't want you to know that they don't know what you're talking about. Okay? So they're not going to tell you that they don't get it. And if they don't get it, do you think they're going to follow what you're asking them to do for their care or their medication or anything else? No. So if you want better compliance, make sure you explain it in a way that they understand it. It doesn't mean you're not dumbing it down. That's not what I'm saying. Don't make them feel stupid, okay, because they will not come back and they will not comply. You just need to explain it in a way that they understand what you're talking about. You know why most of your patients, your elderly patients, don't drink water? They don't usually tell you because you don't take the time to ask them. And when you're trying to draw blood and their veins are rolling away, they don't want to pee all night. That, as simple as that. But maybe if you explain to them, if you drink water, you won't get UTIs, urinary tract infections. You'll be less likely to get sick. They might comply better if you explain to them why they need to drink water. I know you don't want to have to get up 10 times during the night, but just putting it out there. All right, so phospholipids. This is also a lipid. We still have a glycerol backbone. That's the pink part right here. But this time we only have 2 fatty acid chains. That's this part right here. And instead of the third fatty acid chain, we have a polar phosphate that gets stuck on there, and that's the yellow and green part over here. Did you notice what I said? Lipids in general are what? Non-polar. But what I just said is we're sticking a polar head on it, because look at all the oxygens right there on this phosphate group. It makes it polar. So now we have a polar end and a non-polar end to something called a phospholipid. So that phosphate gives you a polar end. Okay. The polar end allows you to interact with what? Right, allows you to interact with water. Okay. And the nonpolar end allows you to interact with what? Nonpolar stuff, fatty stuff, right? Other fatty molecules. So that's an end. Nonpolar end allows interaction with non-polar molecules. Okay, so essentially, if I drew the picture like they've kind of shown you on the map here, and we have like more of a cartoon way of depicting this, here would be the polar head, and then they kind of have their glycerol backbone, and then they have their fatty acid chain like that. But usually the way you'll see it, because that's a little awkward to write it, and we get lazy when we do it, you'll usually see a phospholipid depicted as a guy with a head and two legs on it. Or sometimes it'll be like a guy with a head, and they kind of show you a kink and a straight leg. They're just trying to show you have fatty acid chains hanging off of it. So that's what a phospholipid would look like. So this molecule, this phospholipid, ends up making a phospholipid bilayer. Bi means two. So we have a double layer, okay, and it ends up making the construction of all of our cell membranes. So when I tell you, you are what you eat, the fats you ingest into your body are going to end up being the fatty acids that end up getting put on to your phospholipid bilayer. And it changes. Things move around and stuff. But basically you would end up having your heads here and the non-polar fatty acid chains in the center of that bilayer. I always want to say like Bifrost, right? For Thor fans. All right, so if this is my cell membrane, okay, and let's say it continued on, which side do you think water would be on? The polar side. So you'd have water on the inside of the cell, and you have water on the outside of the cell. And you're preventing too much water from easily getting in or out of your cell, because that's important later too. So just touching on the tip of the iceberg there, introducing it now, you're going to see it again later. Let's see, steroids. Look, these guys are lipid-based. So steroids are also lipids, and they are based or built from cholesterol. And you know how everybody says, cholesterol's bad for you. No, you need it. You have to have cholesterol, but too much or too little of something is not good either. So you need the right amount of cholesterol, just not too much. And we need to make cholesterol because we need cholesterol to make various steroid-based hormones. Okay. And also it is a plasma membrane stabilizer. Plasma membrane or cell membrane. Plasma membrane stabilizer. So hormone-wise, anytime you hear the word or the suffix sterone, you know that it's a steroid hormone. Anybody want to take a guess at a steroid hormone? Testosterone, yes. Testosterone. Sterone. Aldosterone. Progesterone is one also. It's just sterone is in there. How about estrogen? Yeah, it just doesn't have the word sterone in it, but it is. So estrogen, testosterone, progesterone, aldosterone, all steroid-based hormones. you didn't have cholesterol, you would not be able to make these hormones. You wouldn't feel like a woman or feel like a man, okay? You'd feel like something else. All right, so testosterone, progesterone, okay? So let's see. And now let's look at protein. So we looked at carbohydrates, we looked at lipids. Now let's look at proteins real quick. So proteins are organics that also contain CH and O, carbon, hydrogen, oxygen, plus nitrogen, new kid on the block, right? No, I never liked that band, just so you know. Amino acids are the building blocks for proteins. So I abbreviate amino acid as little a. Okay, those are the monomers for proteins. Okay, there are 20 common types or forms that make up all of your proteins. So, for example, we have 26 letters in the alphabet, right? And the way we rearrange those letters depends on the word and thus the meaning of the word. So, for example, if you had the word two is different than the word toe, correct? Same letters, they're just arranged differently, but they have totally different meanings, right? Proteins are the same way. So you have 20 amino acids, and depending on the way that they are linked, depends on what protein it will be, thus the function that that protein does. And if there is one thing that is different, just one thing can cause the difference, for example, between having a normal red blood cell and having sickle cell anemia. Just one. One difference. That's it. So if a protein will not be a functional protein if it has a mistake, like in the DNA coding that tells you what that protein should be. If there's a mistake, it won't make the right protein. So, for example, if I said M dash, what would my dash mean, by the way? Like a covalent bond, right? And I said T dash S dash M dash Q. Is that the same protein? No. Might have all the same amino acids if we're saying these represent amino acids, but it's not the same. So we have our covalent bonds. Amino acids are covalently bound to each other, and that's what gives us Oh, there's a special name for covalent bonds in a protein, though. It's still a covalent bond, but because it's a protein, we've decided to give it a special name, which is peptide bond. That's it. So a peptide bond is specifically a covalent bond between amino acids. So peptide bond. Okay. And let's see what else. Oh, the thing that you need to memorize. So you don't have to memorize all 20 different amino acids. That would be, you're in a biochemistry class. In this class, I need you guys to understand that the term amino acid, what does acid of amino acid mean to you? It has an acidic end to it, right? That makes sense. So here's our acidic end of a generic amino acid, and the amine part of amino has an amine end. That's where the nitrogen of a protein comes in, that end that we talked about here, makes up the amino end. So amino acid, it means it has an amino end and it has an acid end. That's the main thing to know. When you link a whole bunch of them together and you have this sequence of a protein, you still have one end that's still an amino end and you still have one end that will have an acid end on the ends, okay? And if you look at this peptide bond being formed, what kind of reaction is that? If we're linking two amino acids together, we are synthesizing, thus it's a dehydration synthesis because we formed water and we linked them by covalent bond, more specifically called a peptide bond. Thank you very much. And let's see, if you look at this picture here where we've linked a whole bunch of them together, the little purple boxes in here, they're just showing you all the peptide bonds that they form to link it together. Your protein is usually hundreds or more amino acids linked together to create one protein. So it's tons of them together. And there are levels of conformation or structure to a protein. And you just have to memorize that there are 4 of them: primary, secondary, tertiary, and quaternary. So a primary structure is just the linear sequence of amino acids. This is like the linking of the amino acids together. So like the word, right? So this would be like G, A, L, C, V, Z. And if I wanted a really cartoony picture of it, it might just be, you know, like a line. It's just the linear sequence. It's just the order of the amino acids. They are all linked. Amino acids are linked by the peptide bonds or covalent bonds, right? And that's all that the primary structure is. Let's see. And I said, you know, if I had mixed up those letters, it would not be the same amino acid or protein, right? Because it would be a different protein then. And so primary structure is just the sequence or order of the monomers. Proteins are made in a ribosomal factory, which we'll learn about later, and they string the amino acids together, creating those peptide bonds. And when it leaves the factory, it will not stay in this linear formation. Once it leaves the ribosome, it then starts to form hydrogen bonds, which will give it one of two shapes, which is now secondary structure. It'll either get an alpha helix shape or a beta pleated sheet shape. So once it gets into one of those shapes, because it's left the ribosome, it's now in the secondary structure. An alpha helix kind of looks like this. And a beta pleated sheet looks like this. So I like to say you went from primary structure to either an alpha helix, or you went from a primary structure to a beta pleated sheet, and those are all secondary structures. Okay? The reason that they get into these structures are because of hydrogen bonds. So remember, we said hydrogen bonds are a weak interaction, but they're important. So how do we show a hydrogen bond versus a covalent bond? Hydrogen bonds are dotted lines. So it would be like this, dot, 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 holding those coils, or dot, 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 holding the beta pleated sheets here. Okay? So it's secondary is all due to those hydrogen bonds. Now they show you in a cartoony picture here. This one is the alpha helix. This one here is a beta pleated sheet. So those are Greek letters, the alpha and the beta there. Okay. And what else do I want to tell you? From there, after secondary structure, there end up being still other polar areas that can still form more hydrogen bonds or other covalent bonds and things like that. And it can further fold the protein again into more of another shape. And that's when we get tertiary structure. So here's tertiary. And basically, it would be like having your alpha helix fold up. And instead of just having hydrogen bonds that were part of its secondary structure, now it can form hydrogen bonds across to other areas. So it's holding it by these hydrogen bonds and other bonds into a different shape now. So you could either have alpha helix only or beta pleated sheet only, or both could be occurring in a tertiary structure. So it could be a beta pleated sheet now that folded in and created an alpha helix on another area. Okay? And so this is tertiary structure. And in tertiary, you could have a globular protein or a fibrous protein that I'm going to talk about in a second. So if we look at quaternary, so once you have a tertiary structure, this is one polypeptide or one protein essentially. So when it says quaternary, it means two or more polypeptides. So it means two or more tertiary structures are bonding together. And typically it's not just hydrogen bonds, but you'll also have covalent bonds for sure. So this might be, here's your alpha one here that had its hydrogen bonds going on in there, right? And then maybe you have another structure here that's now covalently and alpha and hydrogen bonding to another one. So you have a tertiary structure here and a tertiary structure here coming together to form a quaternary structure. And an example of a protein in our bodies that has quaternary structure is hemoglobin. So it's actually four polypeptide chains, because I said it's two or more, and it's actually four different chains, four different tertiary structures bound together that form a huge molecule, hemoglobin, that holds on to the heme group, which is an iron component, and oxygen, and this is what transports your oxygen around in your body. So it looks like I better stop now, and we will continue finishing with the fibrous and globular protein shapes when we come back on Tuesday. And if you're in lab and you have not, if you're one of the internet students that did not talk to me because you have not been logging in, you need to talk to me, you need to log in. If you don't know how, you need to contact me. And for lab, if you had questions, stay after and I will answer those as well.